Okay, so this is going to be the first recording for the central nervous system. There's going to be a number of recordings, possibly around 10, in which they will be included in the description. It's probably better to keep these at around 30 minutes each, and there will be a lot of recordings, but I think it would be better for learning purposes rather than to have three large, hour and a half videos. So the book I'm using is Human Anatomy and Physiology by Marieb and Hoehn. And this will be the ninth edition. I realize there is a 10th edition, but there's not that much of a difference within, the actual information of the book. So you guys will see that too. Credits to the book will be in the description. The CNS, the central nervous system, the two components to it is the brain and the spinal cord. And from chapter 1, if you guys learn all the different body cavities, the brain and the spinal cord would be occupying the dorsal body cavity. Quick definition, just the CNS is the integrating and control center of the nervous system, in which where high decision making actual choices are being made for the control of the body is within the CNS. Discussing the brain regions would be the two cerebral hemispheres. This is a mid-sagittal section of the brain, so here you only see one cerebral hemisphere, but you're going to have two cerebral hemispheres, and then you're going to have your diencephalon next, which is all in purple. This diencephalon will consist of your hypothalamus, your thalamus, and your epithalamus. We'll get to that probably in maybe part 3 of the recordings. And then the brainstem, which is colored just in green here, is going to consist of your midbrain, your pons, this little bump right here, and then the medulla oblongata right here. And last but not least, the cerebellum here. And these would just be the brain regions, of course, in which the CNS also includes the spinal cord just below. When discussing any system, really, what's also a good way to start is discussing actual development, because it helps you understand and learn all the different structures that you'll be discussing for the rest of the chapter. So I like to start off with a little bit of development. So as for brain embryology, how the CNS starts is through something called the neural tube right here, which they have illustrated right here. The neural tube has its central neural canal here. It's not illustrated, sorry, it's not labeled here, but this would be the neural canal here. So our neural tube has an anterior portion here and a posterior portion. And then rostral would mean actually a snout. So it's just another name for anterior. And then posterior, the caudal portion, or caudal meaning towards the tail. Just another name for posterior or dorsal. So this neural tube is by week 4. It's formed by week 4. And what's going to happen is very quickly, it's going to enlarge in certain areas, and it's going to constrict in certain areas. And then it's going to make our primary brain vesicles. So these primary brain vesicles, in which we're going to have 3 of them right here, this happened very quickly. This is still basically within week 4. So these 3 different primary brain vesicles are going to be your prosencephalon, or your forebrain, your mesencephalon, your midbrain. And when looking at the mesencephalon, you can see that it actually stays consistently as the mesencephalon the rest of the way, just making your midbrain. And then your rhombencephalon right here, which is your hindbrain. A little thing I want to add is these primary brain vesicles, the area that in which it enlarged and constricted was only the anterior portion right here. The posterior or the caudal portion, if you follow this down, it just becomes the spinal cord. So this whole area, all the brain regions are just from the anterior portion of the neural tube. And as I said, this was by week 4 here. So by week 5, what's going to happen is now these structures are going to once again form, they're going to divide and constrict to form now secondary brain vesicles by week 5. So by week 5, we're going to have our prosencephalon is going to divide into 2 different structures. It's going to be the telencephalon and the diencephalon. As we said already, the mesencephalon is just gonna, it's not gonna divide at all, and it's just gonna become the midbrain portion of your brain stem. And then the rhombencephalon, or the hindbrain, is going to constrict in this region right here and make the mesencephalon, and then the myelencephalon. If there's like a little trick you guys want to like know, just to maybe memorize this, this is at least what I did. It sounds dumb, but I just thought of like the first letters here. So TD, as in like touchdown or whatever acronym you want to think about, or mnemonic. But TD here, and then the rest of the M's are actually in alphabetical order, if that helps you in memorization for these different secondary brain vesicles. So the first one here, the telencephalon, we notice the color here is kind of a beige color. We notice for the diencephalon it's a purplish color. And for the beginning of the chapter, these colors stay consistent within the development, and then a little bit in the beginning. They just want to help you. I'll give you an example here in which they're showing the telencephalon in beige, the diencephalon in purple, the entire brainstem in green here, the cerebellum in red, and we can kind of see that consistently within the beginning of the chapter, in which those areas stay within those colors, so you can kind of identify those different structures. So next, no, sorry, we were at the telencephalon, and the adult brain structure that will form from the telencephalon is actually our cerebrum. The cerebrum consists of the two cerebral hemispheres, and these cerebral hemispheres, they have superficial cortex, a deeper white matter, and then something called basal nuclei within the white matter. But this is something that we'll discuss probably in part three. In three, the actual cortex we'll get to a little bit in this recording, and then in the next two. Next would be the diencephalon, in which the diencephalon will consist of the thalamus, the hypothalamus, and then the epithalamus, and then it also includes the retina of your eye. The retina of your eye is also included within the diencephalon, and then mesencephalon, we already said, just becomes the midbrain of the brainstem. Then the mesencephalon here is going to eventually become your cerebellum, and then your pons. And one thing I want to add is, these entire areas are always basically at the same level. So like, let's say we're looking at the pons and the cerebellum of your mesencephalon. If I go over here, you can see that the pons and the cerebellum are at the same level. So that might help you kind of remember the different structures, and when you're memorizing, the locations. And then we have last here, our myelencephalon is going to make the medulla oblongata portion of your brainstem. And then, as we discussed, the caudal portion just becomes the spinal cord. And then remember this neural canal that we were speaking of here. So this like entire hollow cavity here, filled with fluid, it's going to enlarge in like 4 different areas, and that will eventually become ventricles, and then certain like channels or canals, which they have discussed right here. You could look at it if you want to pause the screen, but I'm more so going to be discussing this when in a couple slides when we're discussing the ventricles, and I'll actually come back to this just to connect it all together. We left off at week 5 when we had our secondary brain vesicles here, and during week 5, what's happening here is just our telencephalon and our diencephalon. They're angling towards these 2 flexures right here. So there's one right here, and there's a flexure right here. This one's called the cervical flexure. This one will be the midbrain flexure. And that's really what's happening during week 5. Just the telencephalon and the diencephalon are just angling towards those 2 flexures. By week 13 here, we're going to have our telencephalon here is going to be growing posteriorly and then laterally. So posterolaterally. And that's indicated by the arrows, obviously. And while this is happening, it's going to grow backwards and actually enclose our diencephalon here, starting at week 13. And then also our anterior portion of our brainstem is also going to be enclosed. Not fully enclosed. You can actually, if you were to grab one portion, if I can go right here, like let's say you dissected a brain and you grabbed this portion of this cerebrum right here and then the cerebellum, you can actually pull them apart and you can see these portions right here of our midbrain. So it's not, what's really enclosed is the diencephalon and then a little bit of the anterior portion of our brainstem. So that's really just what's happening during week 13. By week 26, you can see basically most of our adult brain structures are being formed here. So what happens is it started out as the telencephalon as very smooth, but what happens is that as it grows, it starts to fold and crease, and it makes these convolutions here. So these convolutions are going to increase the surface area of our cerebral cortex, the superficial, the more outside portion. And what's going to happen is you only have a limited amount of area that you can occupy within our skull. So those convolutions, these foldings, can only be beneficial in which we're going to have more area to keep more neurons within a limited amount of space that we occupy. So this is very beneficial to us. That's and then we're going to kind of leave off here when it comes to brain embryology. We can see the different adult brain structures here, which we have our telencephalon, diencephalon, and then our midbrain here, pons, cerebellum, and medulla oblongata, and then our spinal cord here. And then, as I said, now we can discuss finally the ventricles of our brain, in which, remember, the neural canal, that hollow space filled with fluid, will become these ventricles here. They're just like little central hollow cavities here. They're filled with fluid, and this fluid, they actually call it CSF. It's cerebral spinal fluid, in which it's coursing all throughout these ventricles and also on the outside portion of the CNS. We'll see that in probably 2 slides from now. But so these ventricles are they came from the neural canal, from the central portion of that neural tube. So when discussing these ventricles, you learned in chapter 11, just the previous chapter, the different neuroglia. Those different neuroglia from the chapter 11, we learned about the ependymal cells, in which in the CNS, the CNS is lined by ependymal cells. I put a little picture here. So they show here brain or spinal cord tissue on the bottom. So this would be our CNS here, in which it's lined with these ependymal cells. And remember, these are ciliated cells here. So we're going to have our CSF here, and then they're going to help to kind of distribute, kind of move along that CSF within the, within the cavity it's occupying. So each of these ventricles have these ependymal cells lining it, and quickly we can just discuss each kind of ventricle. The first one is these C-shaped ones right here. These are the lateral ventricles. Remember, lateral means away from the midline. So these, just a little anatomically, you have 3 horns. You have your anterior, posterior, and our inferior horns of our lateral ventricles. So we're going to see that right here, there's a little bit of a division here, in which they're not continuous with one another, but they are divided right here through a very thin median membrane. It's called the septum pellucidum. So this septum, whenever you see septum, means wall. So that's just a little indication that you're seeing some kind of a wall there. So the septum pellucidum, only anteriorly here in this portion, divides those lateral ventricles. And remember, all these ventricles are continuous with one another. So this lateral ventricle is going to be continuous with the next ventricle, and then this ventricle is going to be continuous with the next, and then eventually the rest will be continuous with the outside of the CNS, which will be discussed. I'll even show you a picture, I think, in the next slide. But the way in which this lateral ventricle is continuous with the third ventricle, remember there's 2 of them, so it immediately goes to just a third ventricle, is through this little channel here. It's called the interventricular foramen. So you can see these channels going through here, the interventricular foramen, and it passes eventually into the third ventricle. So this third ventricle is going to be in the area of the diencephalon. So remember, the diencephalon was our purple region. We had our hypothalamus area, we had our thalamus area, and then our epithalamus area. So this third ventricle is going to be occupying the diencephalon. And then once again, this diencephalon will be continuous with our fourth ventricle right here. So it's continuous with the fourth ventricle through this little canal here. You can see it a little bit better right here. This little kind of short canal called the cerebral aqueduct. This cerebral aqueduct is going to be running through our midbrain. and then eventually this cerebral aqueduct will get into our fourth ventricle. So real quickly now I could kind of go back here because we're discussing a little bit of these ventricles. We discussed how the T-encephalon, which was our cerebrum, it had those lateral ventricles, the C-shaped ventricles. And then we discussed how it went through the interventricular foramen and then eventually went to the diencephalon, in which had the third ventricle. And then we saw how it went through the midbrain and then through that little canal called the cerebral aqueduct. And then from there it went into the fourth ventricle, which is in the area of the myelencephalon and the mesencephalon, through that pons, cerebellum, and medulla oblongata. And then anything below that is just the central canal of the spinal cord. So I don't really have to go back to here anymore. I just wanted to show you that real quick. You can see our central canal right here, through our spinal cord right here, and then our fourth ventricle right here within the pons, cerebellum, and medulla oblongata. So now we reach our fourth ventricle here. And then in this fourth ventricle, we're going to see these little kind of like walls within the roof. Because think about the fourth ventricle ending about this region right here. So there's not really too much happening here, but in the roof here, we have these little kind of walls here. So on the walls, we have kind of things marking those walls, in which they're called apertures. So here we have our lateral aperture right here, and then marking the side of the walls, and then we have a median aperture right here. The way I pronounce it is actually pretty funny, sorry. So the median aperture is right here, and then lateral right here on the side of its walls. And you can see these little holes here, in which now, remember, these are all continuous with one another, but not through these holes, they can actually be continuous with the actual outside of the CNS now. So, blood is, I'm sorry, fluid is going to leave this, the ventricles, the CSF, and then eventually get into the outside of the CNS here, through these different apertures. What they're going to eventually get to is something called the subarachnoid space. The subarachnoid space is a fluid-filled area that surrounds your CNS. I'll show you actually right here, because we're basically done with this picture here. So here we can see that by the usage of these apertures here, they were able to leave through these holes and then eventually go to this subarachnoid space right here. So this is all subarachnoid space, the area surrounding the CNS here, in which we have this CSF also there. And the way in which they were able to get there was through these the roof of the fourth ventricle, going through these little holes right here, eventually into the subarachnoid space. And you can see indicating through all these arrows that all this fluid is continuous. You can see it going from the lateral ventricle here, eventually to the interventricular foramen, to the diencephalon, in which it's the third ventricle, through the midbrain right here. This is the midbrain. You can see it's going through the cerebral aqueduct, then into our fourth ventricle. We can see the different apertures here, the lateral and the median apertures here of our roof of the fourth ventricle. Eventually, it's going to let it be continuous with the subarachnoid space of the CNS. You can see the subarachnoid space here, surrounding also the spinal cord. And then even here, the fourth ventricle just becomes continuous with the spinal cord through its central canal here. Okay, I guess the next component to speak about now is our cerebrum. This portion here, which comprises basically 83% of the mass of the brain. So a huge portion of that. So that cerebrum, it consists of two cerebral hemispheres, as we discussed. And you can see a lot of foldings here. We saw within week 26, those foldings started to occur within development. So the different portions of these convolutions, we actually have names for them. So we can see a little piece here taken out here. And just the individual little ridges here of elevated tissue, they call it gyrus, or gyri for plural. And then when you're separating these different gyri, these different portions of the little foldings here, let's say like right here, they actually call it a sulcus here, dividing these different gyri here. So it's very shallow groove. It's not too deep. When you have a very deep groove right here, a very deep sulcus, they call it actually a fissure. And so right now let's just discuss really the sulci here. So a couple, sulci that to discuss. One will be the central sulcus here. The central sulcus is located right here, and that will be dividing our frontal lobe here and from our parietal lobe here. So we're gonna have anteriorly we're gonna have our precentral gyrus, and then posteriorly we're gonna have our postcentral gyrus. These areas we're gonna talk about when discussing motor and sensory functions of our cerebrum within part 2 of the chapter. Next we have our parieto occipital sulcus right here. So this will be dividing our parietal lobe from our occipital lobe. And then we have our lateral sulcus located right here, in which we're going to have our temporal lobe divided from our parietal and our frontal lobes. Okay, so next we talked about the fissure, which is very deep, kind of a much deeper groove than the sulcus was. And this is going to separate large brain regions. So two very commonly discussed ones are the median longitudinal fissure. You can also sometimes see just as longitudinal or just as sagittal fissure. Also, that works too. Here they just have it as the longitudinal fissure here. But this is going to be dividing our two cerebral hemispheres from each other. You can see it's very deep right here. And then our transverse cerebral fissure right here. Sometimes they even just call it the transverse fissure, but it's going to be separating our cerebellum from our cerebral hemispheres. So those are two very notable fissures there. Once again, I put the little diagram here in which the fissure is a very deep sulcus, as you can see here. Very quickly, when it comes to the cerebrum, I'm sure you've seen the different lobes. So the 5 lobes of the cerebrum is going to be our frontal lobe here, our parietal lobe, our occipital lobe here. So the pronunciation, it could be occipital. I just say occipital. And then the temporal lobe here. They do say that there's a 5th lobe here, actually located deep to the temporal, parietal, and the frontal, which are covering it. So this insula here is considered our 5th lobe here. It's just another area of cortex, sometimes called the insular cortex. We're going to discuss all the different functions, I think, in the next or third recording. So then the cerebrum. Talking about the cerebrum, there's a certain pattern that follows here. We can see different regions in which we have our outer region, our more deep, kind of whiter region, and then these different colors they indicate here. So the most outer portion is going to be called our cerebral cortex here. The most superficial portion. This superficial portion, the cerebral cortex, is a lot darker here, and it's actually composed of something called gray matter. So that gray matter is going to be actually that kind of darker color because it's composed of neuron cell bodies, dendrites, associated neuroglia, and blood vessels. But what you're not going to see here is any fiber tracks. That fiber tracks you're going to see within the white matter actually. So that would cover our cerebral cortex. The inner, more deeper here portion, the white matter here, it's actually going to be consisting of myelinated fibers or axons. And remember, when you have anything myelinated, it's actually kind of like a fatty kind of substance, in which that would indicate the reason why it's white here. So it's a bunch of myelinated axons here in the white matter. So, and they're bundled into large tracks. In chapter 11, you guys discussed tracks, nuclei, things like that. When nuclei were clusters of cell bodies within the CNS, and then tracks were clusters of axons within the CNS. So that's the reason why they call them tracks here, because it's the CNS, in which is a bunch of fibers bundled together. And then the next thing we're going to discuss is the basal nuclei here, indicated by the pink here. So these basal nuclei are, I call them subcortical, which means just below the cortex. So they're actually gray matter, and they're not located in the cerebral cortex. They're actually located deep within the white matter. So these basal nuclei are going to have a certain function also. That's going to be actually in part 3 of our recordings. So the pattern of the cerebrum, you're going to have the outside cerebral cortex composed of gray matter, the white matter just deep to that, and then gray matter within, actually located within the white matter, called our basal nuclei. The cerebral cortex, is the next thing we're going to discuss, in which this area has a high function. So here I have as whereas part of it's where our conscious mind is found. So this is going to allow us to communicate, remember, understand, initiate voluntary movements, and be aware of ourselves and our sensations. So this is a lot of a higher kind of thinking, is the function of the cerebral cortex. So here it contains billions of neurons in 6 layers, 2 to 4 millimeters thick. And depending on the book you guys use, it could be 2 to 4 millimeters thick. Some say 1.5 millimeters to 4.5 millimeters. So it just depends on the book. A lot of these numbers are it just depends on what book you're actually reading. So and then actually the cerebral cortex, although like here we're looking at where it doesn't seem like taking up that much room, in which the white matter looks like it's more of the higher amount of mass. The thing is, it actually composes the 40% of the brain mass. So the reason that is because it's highly folded here. So these convolutions actually triple the surface area of the cerebral cortex. So that would explain the reason as to why it's 40% of the brain mass. Also, the cerebral cortex, there's something called lateralization, in which you have a specialization in which a certain part of the cerebral cortex will be responsible for controlling a certain part of the body. But the thing is, when you're discussing, let's say, the left cerebral hemisphere, what it's actually going to control is something of the contralateral side, so the opposite side. So if we discuss the left cerebral hemisphere, it actually controls motor and sensory controls motor functions of the right side of the body. While let's say the right cerebral hemispheres, it gets impulses from the left side of the body for like sensory, and then it controls the left side of the motor functions. So it's actually it controls the contralateral side, the opposite side. So the right will control the left side of the body, and then the left will control the right side of the body. Also, when discussing the cerebral cortex, everything in the cerebral cortex, they're all interneurons. So there's no motor or sensory neurons in the cerebral cortex. So you're going to see those motor and sensory neurons within the white matter. So they're going to synapse with those interneurons of the cerebral cortex. Some kind of decision or some kind of decision or choice is going to be made, and it's going to be sent right back to the white matter. So all cerebral cortex neurons are interneurons. And then last year the cerebral cortex consists of 3 types of functional areas. So it's going to consist of motor areas, sensory areas, and then association areas. And that will actually be the part 2 of this recording. We'll be discussing all the different functions of the cerebral cortex, because there is a bit of a lot. So um, eventually I'll have the links up in the description once these videos start to appear. So that would be it for this lecture.